add glucose, we could add maltose, we could add sucrose, fructose, starch. I mean, we can basically add any organic molecule we want, and as long as that organic molecule plugs somewhere into the glycolysis pathway, then we would expect fermentation to occur. So think of, and this is really only, this is going to help if you've seen the virtual lab. If you haven't seen the virtual lab, you're going to need to think extra hard, I guess. What's one way that we can measure, just using the materials that we have in class, what's one way that we can measure the rate of fermentation?
A clock. Fluid of oxygen consumption?
Okay, we can look at oxygen consumption, but that is actually not going to work because why? What do we know about fermentation? It doesn't need oxygen. There is no oxygen consumption for fermentation. Glycolysis is anaerobic, and all fermentation is doing is it's regenerating NAD+ so that glycolysis can continue. Okay? So we can't really do that, especially, well, here. What do you think?
Glucose consumption?
We can look at glucose consumption, so we can look at the rate of, or I'm sorry, the change in maybe glucose concentration. So maybe if we took those glucose test strips, we can use a test strip every couple minutes to look at how much glucose is being consumed. That's something that we can do. That wouldn't necessarily tell us, though, whether or not fermentation or cell respiration is happening. Because glucose is still going to be consumed if these yeast are respiring aerobically.
Weight difference, because it builds lactic acid, doesn't it?
Okay, you're onto something. You're onto something. First, let's make sure that we understand what the yeast are making. There are two products that can form as a result of fermentation. One of them is lactic acid. We know that yeast is used to make bread. What is it about yeast fermenting that's so valuable in the bread making process, Victoria? Carbon dioxide. So they're not making lactic acid. Producing lactic acid from pyruvate does not make any carbon dioxide. So what must they be making? Carbon dioxide. That accounts for one of those carbons. So one carbon is in the form of carbon dioxide. What about the other two? Ethanol. The other two carbons are found in the organic molecule ethanol, which is just beverage alcohol. Okay? So in this case, we know that they're going to be making carbon dioxide. So how can we use that knowledge to help us figure out exactly how they're fermenting, or the rate that they're fermenting at? Are they consuming carbon dioxide? They're actually producing carbon dioxide. So we can look at the carbon dioxide production here. Okay? So that's my water, Y for yeast, and S for substrate. So if this is an open container, how are we going to actually measure carbon dioxide production? Isn't the carbon dioxide just going to leave the test tube into the atmosphere? How are we actually going to measure how much carbon dioxide gets made? What's something simple we can do to capture that carbon dioxide? Huh? We could put a balloon over it. Okay. We could take a balloon and stretch it over the top. What's going to happen to that balloon over time if carbon dioxide is being produced? It's going to inflate, right. Now, in the virtual lab, I just want to reemphasize this point. In the virtual lab, when organisms expel carbon dioxide, I'm sorry, let me backtrack. When organisms consume oxygen, they're taking gas out of the atmosphere. But the reason that we're not seeing any change in pressure in that system is because why?
It's closed off.
Well, it is closed off, but importantly, they're replacing it with carbon dioxide. So if I take an oxygen gas out of the air, but I replace it with a carbon dioxide gas, I've done nothing to change the pressure of the system. So I can't measure pressure changes. Here, we're only going to see an increase in pressure if fermentation is happening, because all we're doing is just adding carbon dioxide. We're not taking oxygen away. Does that make sense? That's why we don't have to add anything to this to remove the carbon dioxide. Because they're not using any oxygen, they're just adding carbon dioxide. And where's that carbon dioxide ultimately coming from?
The breaking down.
The breaking down of what?
Pyruvate.
The substrate, yeah, whatever the substrate is. Ultimately, it's the breakdown of pyruvate, because that substrate is going to form pyruvate, pyruvate will break down. So when someone goes on a diet and they say that they want to burn fat to lose weight, what does that mean? Like when they say they're burning fat, what is actually happening, Valeria?
They regulate their diet so they have like low glucose levels.
Okay.
So that your body starts using up the fat.
Okay.
Doing glycolysis and the whole ETC cycle.
Okay, so low glucose levels are going to promote the breakdown of stored organic molecules. And when they say that they're burning fat, what's literally happening to that fat? I don't know if you said it before or not. Say it.
It's broken down.
It's going to be broken down
Like
in what process?
glycolysis.
Glycolysis. But remember, some fats plug into where? Right. So they're going to enter that pathway either by plugging into glycolysis or plugging into acetyl-CoA, or if we're talking about some amino acids can plug directly into the citric acid cycle. Your body typically doesn't break down amino acids if you're on a diet. If your body is breaking down amino acids, that means your body is eating its own muscle, and that's only going to happen if you enter into starvation mode. So when you're burning the fat, where is it going? It's not disappearing. Where's the fat going?
Heat.
It's not being released as heat. I mean, when you break down fat, there is heat that's released, but the actual fat molecules aren't being converted into heat. They're still there, just in a different form. Where are they? No, they're not in ATP. The energy from their breakdown is used to make ATP, but I'm talking about like an actual fatty acid. So maybe like C8H22O3, I don't know.
Is it in your blood?
That was stored in fat tissue. When you burn it, where does it go?
Is it the adipose? Is that what it's
Adipose tissue is the fancy name for fat tissue. Yep, the white fat
Where does
tissue.
it go?
Where's the fat molecules going? If you're burning the fat, where are they going? In the form of, specifically, carbon dioxide. Yeah, they're being converted into CO2 and water, and you're literally breathing that out of you. That's how you're losing that weight, is you're converting it into gas and you're breathing the gas out of you.
So if you breathe a lot?
You're not you're destroying the molecule, but you're not like destroying the atoms. The atoms have to still be accounted for. They're just being converted to gas, and you're exhaling the gas. You're losing weight by exhaling gas. That's why hyperventilating is such a good way to lose weight. Try it sometime and tell me how it works.
Wait, what does
Yeah.
hyperventilate mean?
Don't do
Oh,
that.
over like with that bag?
That's not an effective way to lose weight. That's an effective way to pass out. Don't do that.
Yeah, I think it's gonna be.
All right. Okay? So here's the question though. How are we going to quantify this? What does quantify mean?
Increase?
Numbers. Put it in numbers. Like, sure, we could say that the balloon is going to inflate and that it inflates more as time goes on, but that doesn't give us any sort of quantitative information. How are we going to convert this into some quantitative data?
Take a diameter of the balloon every once in a while.
Okay, so this is a sphere. Let's just measure the circumference. But I mean, yeah, you're exactly right. Let's just take like a little measuring device, wrap it around the balloon every minute, and see how large the balloon is by measuring its circumference. And the idea is that over time, the balloon grows in circumference
The
as
reaction.
long as carbon dioxide is being added. And then we can get actual numbers that we could chart on a graph and compare. So maybe we run different experiments that have different substrates, initial substrates. What? Yes?
We take a small sample of the gas in there somehow without letting it all out and find some way to measure it to see how much carbon dioxide is in there.
Yeah, I mean, you're on the right track. What you can do with that, if we have these, I don't know if we do or not, you can use what are called pressure probes. Basically what you do is you have your test tube, you insert like a black rubber stopper that has a tiny little hole in it that allows you to fit a tube into the test tube, and this is a little probe that detects pressure. So it's not measuring the carbon dioxide concentration. What it's doing, it's instead measuring changes in pressure. Again, that's another way that you can quantify it. What do we need to do? Anytime you're ever asked to set up an experiment, here are some of the questions that you should ask yourself. What are we trying to figure out? What are we going to measure? How is that data going to help inform us about what's happening? So here, we're measuring the rate of fermentation. How are we actually measuring it? Looking at changes in pressure, either using a balloon, or if we want to use fancier equipment, a pressure probe. How are we going to ensure that the balloon is increasing in volume only because of fermentation, and it's not something else the yeasts are doing that are causing changes in pressure? Like, what are some controls that we can do to make sure that this data is valid? Someone else who hasn't spoken yet. There's more than one answer. Valeria's already spoken, Victoria's already spoken. Anybody else? Jack, you're still Jack, you're just in a different seat. Abby?
Make a semi-permeable membrane, right?
Yeah, no.
I answered, but never answered correctly.
What would happen if you just didn't add yeast?
There, thank you. What do you think would happen if you didn't add yeast? This is the control.
Please.
What would happen? What would we expect to happen?
Nothing.
Nothing. We would expect the balloon not to increase in volume.
Right.
So this is an experiment that ensures that yeast must be present in order for carbon dioxide to be produced. What else could we, that's one thing we can do. What else can we do?
What was S stand for again?
We did it without any yeast.
Do it
Now
on
let's
these
do it without
Substrate.
any substrate.
Oh.
This is going to verify that what?
The sugars are fine.
Good, that the sugar is required for fermentation to actually take place, that the yeast aren't producing carbon dioxide on their own, which would skew our data. Yes?
Could you do it with a different solute other than water? Or would that not really
You
make
could, that probably, if anything, you would have to do it with some sort of polar solvent, because you want these sugars to dissolve. And then you would have to find a polar solvent that also doesn't kill the cell. I don't know what solvent you would use.
This is totally random, but is milk a polar solvent?
No, milk is composed of water with little fat globules suspended in the water. So like if you were to look at milk at the microscopic level, it's water. And then you have, remember that word micelle? Do you remember what micelles are? Micelles are basically a single phospholipid layer with all of the tails pointed inwards so that it forms
A glob.
like
Is
a
that like
globe,
people say
a globule.
Micellar water? Is that what's in
Cellular water?
There's like this thing that is like a cleanser, and they say it's like a micellar water. And it's,
I don't know what do they say micellular or mycelium? Because mycelium is a fungus. Or do they say like micellular?
do you know what I'm talking about, Valeria?
I don't know, you always have to,
thing.
it could just be a marketing ploy.
Oh,
Like,
I think it's just a marketing thing.
I don't know, I don't know about that.
How do you spell it?
I don't even know what it is.